Good afternoon, everyone. Welcome to the first seminar in the Department of Biological Sciences Spring 2022 seminar series. My name is Dr. Shelley Thomas, and my co-host is Dr. Claude Krummenacher. We represent two committees who are sponsoring this event, the Seminar Committee and the Student Professional Development Committee, and we are very pleased to have you here today. This session is being recorded. So our seminar series includes 5 more seminars this semester. We have 2 research presentations, and you can see from the screen that we have them listed here. Let me scroll down just a little bit. So here we have Dr. Archana Kumari. She'll be talking about hedgehog signaling regulations of taste organs. And we have Dr. Sarah Wright, who will be talking about genetic and life history trade-offs of white clover climate adaptation. And then we also have 3 career seminars. And Ms. Laura Tremonton is the Assistant Chief Forensic Scientist for the state of New Jersey. We have Mr. Merlin Benner, who is a wildlife biologist, and Ms. Kendra Flores is a genetic counselor. And so they will be speaking about their careers and how to get into that career and what it is like being a participant in that career. In today's seminar, we're going to have Dr. Dan Duran. He is the assistant professor and naturalist in the Department of Environmental Science here at Rowan University. He graduated from Stockton University with a bachelor's in environmental science and earned his master's in entomology from the University of Missouri. He was a pre-doctoral fellow at the Smithsonian Tropical Research Institute, and then he earned his PhD in evolution and ecology from Vanderbilt University. He was an associate teaching professor at Drexel University for 11 years before he came here to Rowan University in 2018. And his research interests are in the fields of systematics, taxonomy, and biodiversity conservation. And his research is primarily focused on biodiversity exploration and the discovery of cryptic species. And today's talk is entitled Tiger Beetles as Models for Cryptic Species Discovery. So as you are listening, hopefully you're going to be coming up with questions and thoughts that you want to be expanded on. And so here's the format of this seminar. There will be about 40 minutes of a formal research talk, and then we will go directly into a few minutes of Dr. Duran talking about his career pathway and how he got to where he's at. And then we're going to have a Q&A, a combined Q&A for both the research section and the career part. And so what we'd like you to do is we encourage you to write your questions in the chat. And you'll notice you can only send it to a couple of people. Please send your questions directly to Dr. Claude Krummenacher. And he will be organizing those, and then we will be asking them aloud during the Q&A time. So again, send those questions as soon as you come up with them. Please don't wait until the Q&A time to post those, because we need to organize them and get things together. So it's really beneficial to us if you post them as soon as possible. And if you are doing this for a class, for participation for a class, and you need credit, make sure that you include your instructor's name and follow whatever format or other instructions that they have already given you. So you will not, when you post questions, you will not be able to see everybody else's questions. You will only be able to see your own, but we are receiving them. So again, please post those to Dr. Claude Krummenacher. And I need to move this so that I can stop sharing. And right now, I would like to turn this over to Dr. Dan Duran.
Thank you for having me. This is great. I'm really excited here to talk at Rowan today. And I changed the title by just a couple of words. Instead of tiger beetles as models for cryptic species discovery, I want to go a little broader and go biodiversity science. So we're going to talk about tiger beetles today, their biology and natural history. And many people have never heard of a tiger beetle, but hopefully you'll find them to be pretty interesting after this. And then we're going to talk about using tiger beetles as a model for biodiversity exploration, including the discovery of cryptic species, that is, species which are distinct evolutionary units, but you can't tell them apart morphologically. So that's one of the things that we're going to talk about. And also applied ecology. A lot of basic science, when you do it right, there's applications that can come from this. And I want to talk about some of the really interesting things that we're working on right now. And then finally, I'd like to talk a little bit about rare species and conservation, because whenever you study biodiversity, very often that has a conservation flavor to it. And we'll discuss that. Okay, so how do you know what a tiger beetle is, right? Tiger beetles are very diverse. They're all over the world. There's many different body shapes. Some of them have giant heads and big mandibles, others don't. And so, are they even a real group? One of the questions is, were tiger beetles a real evolutionary lineage? And so we can recently address this now by saying, using entire genomes, we find that tiger beetles do form their own monophyletic lineage, also known as just an evolutionary lineage, an evolutionary clade, a true actual lineage. And so they are distinct from the ground beetles, of which they had often been placed within. And so this is a real thing, and we can talk about them a little bit more and say, still, how do you know that you have a tiger beetle? There are things that are morphological characteristics which do define them and are consistent across all tiger beetles. We could refer to those as synapomorphies, shared derived characteristics, which are unique to that group. And so tiger beetles have at least 3 or 4 synapomorphies, but a couple of them are more obvious than others. One of them is the antennal insertion. The antennae are inserted right between the eye and the base of the mandible. So that's a distinctive thing there. And then, very simply, they have multiple teeth on each mandible. They always have at least 3 teeth on each mandible, and no other group of similar beetles will have that. And so this here is a tiger beetle with an exaggerated labrum. This has a very long front lip, which many tiger beetles have, and we still don't actually know the function of what this does, although we do believe it's probably related to the specific prey that they capture. And so some people believe it's something like a lid of just tamping down on top of a prey item while the mandibles slice it up. Now this here is a close relative. This is a type of ground beetle that, at first glance does look very similar. It's not a tiger beetle. It doesn't have multiple teeth on the mandibles, and also doesn't have the same sort of antennal insertion as well. Now, tiger beetles are fast-running generalist predators, meaning they don't really care what they eat. They'll eat anything smaller than themselves. That could be an ant, it could be a springtail, it could be pretty much anything smaller beetles, and they're found in primarily open, early successional habitats, areas where either wind or water action, fire, storms, something has kept the ground open. And that can also be due to chemical factors such as alkali flats, places where there's a lot of salt and vegetation can't grow. But they do like these open areas because they hunt, and they have to hunt using vision. And so when they hunt using vision, plants get in the way. You have to be able to run fast and catch things and see them out in the open. So that's where you typically do find tiger beetles. This is a classic habitat for tiger beetles. Here we can see this is a big sandy blowout in the Florida scrub. And so a place like this is awesome because it has open areas where they can hunt. It has clusters of grasses and shrubs and trees where they can get into the shade if they overheat. Many places where you find tiger beetles are going to be in these so-called waste areas. They're going to be in places that people don't tend to value very much for biodiversity conservation. It's unfortunate, but people typically think of forest as being worthy of conservation. And areas like this, just open kind of scrubby fields and places with some bare ground, they don't think of those as being high priority. But unfortunately, this is inaccurate. Many species live in these early successional habitats. And it's challenging when you say tiger beetles, because every time you find a good population of something, there's always seems to be a for sale sign. And they also occur in forests too, but they typically are found in the more open habitats within forests. So trails, light gaps from where trees fall down, things like that. Now, there's about 3,000 species found globally. More species are being discovered every year. I've discovered 11 new species since 2013. And they're found on all continents except for Antarctica, and pretty much everything but the most remote oceanic islands. They're not in Hawaii, but they're in a lot of other places. They're in Socotra, they're in Fiji, they're in lots of remote oceanic islands. And anywhere you can find a beach, you can probably find tiger beetles. Now, they have been used as models for studying a number of different evolutionary phenomena, including ecological niche partitioning. So this is an interesting thing where basically you'll find many different species will co-occur in the same habitat, and they all seem to be generalist predators that largely eat ants or other similar prey of that size class. And so the question was, do they actually diverge in the mandible shape and size more than would be expected by chance in order to avoid competition? And the surprising answer is all the research to date so far has said that it's quite the opposite. They have actually converged upon a similar size and shape mandible across different independent lineages because apparently it's just best to get good at hunting the most common prey that occurs in a given community. And so this certainly suggests that resources, at least prey resources, are not limiting. So that's interesting. They've been used in biodiversity conservation studies. It's been shown that tiger beetle species richness actually predicts the species richness of other groups, including butterflies and birds. And so they can be surveyed relatively quickly compared to all the other biodiversity in an area and have been proposed as model bioindicators, species of which you could use to determine the overall diversity at a site simply by measuring them. And so this is an interesting idea and has obvious applications for biodiversity conservation. Now they're very popular. They're very popular with amateurs of all stripes, naturalists, certainly with professional entomologists and others as well. They are well represented in museums and private collections. And one of the reasons why they are, and there's so many field guides written about them, including this field guide that I co-authored in 2015, a field guide to the tiger beetles of the United States and Canada, is because so many of them are absolutely stunningly beautiful. I mean, there's an incredible diversity of colors and patterns, shapes, sizes. And when you see some of these things, they look like artwork. They look like jewelry. You know, it's really incredible. And the question is why? Why are they so colorfully patterned? One obvious interpretation could be that they are extremely toxic. That's been brought up. Some of them are, in fact, quite toxic. However, it doesn't correlate well with gaudiness. The ones that are most brightly colored are often not the most toxic. So this is interesting and a little hard to explain. We find that some of the species which are actually quite cryptic, that look like dirt or stones or sand, they actually are often quite toxic themselves. So again, there's not a clear indication of why some are colorful and why some are not. And you might think that perhaps this is due to sexual selection, where perhaps there's something going on where the really colorful species are engaging in some sort of mating displays. And it turns out that that's not going on either. So we don't have a really good handle on why about 50% of species are so colorful and 50% seem to be rather drab. Now, some of those drab species, you can see why they look this way. I mean, they blend in incredibly well with their surroundings, which makes it very difficult for predators to find them. This is one of our own species, which I will talk about in a little bit as well as a conservation priority. This is the gray ghost of the dunes, also known as Ellipsoptera lepida. And these have such incredible spectral matching to their environment that their shadow is more visible than the actual insect is on its substrate. Another strategy is look like something noxious, look like an ant. You know, that's a great way to avoid predators because many ants, especially in the tropics, can bite hard and can sting hard. And so that's one strategy. Here's a particular species on the bottom of this image, which is mimicking one of the velvet ants in the top of this image with the bright orange spots. And velvet ants are notoriously noxious and one of the most painful stinging insects in the world. And so it's a good idea to mimic something like that. Now, despite these various defenses that they have, they are still prey for a number of different groups, including robber flies, wolf spiders, lizards, actually woodpeckers as well. Some owls will eat them. And so there's this war going on between them and their predators because they're in these open environments where they're highly visible. And so some of them have actually evolved hydrogen cyanide as a repellent, which is a pretty effective repellent, apparently, according to some research that's been done. And those that have that typically have a bright red abdomen, which they flash when they start to fly away, warning any potential predators, I'm extremely noxious. And this works. This is it's enough to repel a kestrel. They create that much hydrogen cyanide. And they're actually one of only 3 groups of invertebrates that can create endogenously their own hydrogen cyanide. Okay, so let's talk about using tiger beetles as a model for biodiversity exploration and applied ecology. Now, when we look at tiger beetles, as I said, they've been very well studied, and the taxonomy has been thoroughly worked on for many years, for hundreds of years. However, there's significant problems with the taxonomy right now, and this is one of the things that I'm interested in trying to address. most of the different species and subspecies are defined based on color patterns and the pattern of hairs on the head and other structures. And a lot of this phenotypic variation, this variation in the observable characteristics of these beetles, it's not clear what significance it has. Some recent research has shed some light on this, but for example, color. Here we can see the species Cicindolidia punctulata. There's blue form that occurs in the southwestern United States in Arizona and New Mexico. And we have a brown form that's right here in South Jersey. It's part of the same species. They do integrate in an area in Texas. Everything east of Texas is brown and apparently the same subspecies. But it turns out this is not a good indicator. This color pattern is not a good indicator of relatedness, because if you were to take blue individuals of punctulata chihuahua from Arizona and you were to rear them under the environmental conditions of New Jersey, they look like ones from New Jersey. So this is clearly phenotypic plasticity. This is not to do with genetics, any sort of long-term break in gene flow between populations indicating a subspecies. That's not what's going on here. And so I've been trying to reevaluate a lot of the characteristics which are used to identify species in the group. And one of the reasons why color is particularly bad as an indicator has to do with the actual developments of the cuticle in tiger beetles. So tiger beetle colors are all caused by a single pigment. We can see here this brightly colored, almost rainbow colored Cicindela scutularis. Its cuticle, if you were to do a cross section of it with SEM, you can see that there's actually dark layers of melanin, and then there's thin layers of clear cuticle in between those. And they're laminated, and the optical thickness of the melanin layers determines the wavelength of light that will be reflected. And so if they're 540 nanometers apart, they reflect green. And so slight differences in the contraction in the drying of these layers during development can actually give completely different colors to the same beetle. It's nothing to do with genetics. And so it's been very frustrating, because a lot of the amateur tiger beetle community, and many of them are describing subspecies and species, are using characters which we know developmentally are the poorest characters to be using. And so the question becomes, how well do these predict relatedness? Do they predict at all? And what should we be using in place of them? And sometimes you'll get black forms of a species that are created when those laminated layers become disorganized and they no longer reflect pure saturated color, and then they look black. So recently I published a paper, Geographic Life History Differences Predict Genomic Divergence Better Than Mitochondrial Barcodes or Phenotype. And we're going to walk through what we learned in this paper here. So first of all, I did say mitochondrial barcodes, and I want to make sure everybody knows what that means. So some may be familiar with this and some may not. But basically the idea is there's a single gene, cytochrome oxidase 1, that can be used to genetically identify all of life. And so the idea is you take this gene and you compare it to other individuals, and when they form a single clade together, a single evolutionary lineage, and those diverge by more than about 2 or 3% from other such lineages, you could call them their own species. And so that has been done especially in groups like birds and butterflies. Here's a group of butterflies. The adult butterflies all look the same. You can't tell them apart. But the caterpillars are different looking for each of these butterfly groups. And these populations, there was a question as to whether or not they were distinct species. And according to the mitochondrial barcodes, each one of them forms its own lineage, which is divergent from others by a couple of percent. And so they have been considered to be cryptic species. We couldn't tell them apart by looking at the adult butterflies, but we did look harder and found some differences, also morphological, also phenotypic, but they were basically identified based on that mitochondrial gene. Now, the question is, how well does this work across all of life? Because it's largely been tested on certain groups like butterflies and birds, but we have reason to believe they may not be the best test examples. So I would like to test this in tiger beetles and see how well it predicts major lineages of tiger beetles as well. And so here is a group that I thought would be a really, really interesting one to study, because it's incredibly variable. This is all considered one taxonomic species prior to my study. And the question is, like, what is going on? How do you identify this group? Well, they have incredible variation in color and pattern, but there are phenotypic characteristics which are consistent across all of them. They all have a pronotum, which is the area behind the head, that is shiny. It's glabrous, meaning it doesn't have hairs. There's other characteristics. We won't bore you with the details of them, but these were all considered to be one species. And they all have a similar habitat type. They all live on limestone outcroppings. And so the question was, are all of these different color forms different? Subspecies? What's going on here? And so I wanted to look at this and figure out in an area where we have extreme phenotypic developmental, potential developmental plasticity, let's see what's going on with the real evolutionary lineages. And so we were able to get mitochondrial data from many, many, many different individuals throughout the ranges of all of the different subspecies, all of which are shown by a different shaped icon. And we were able to get nuclear data from many of them as well. And so what we found was they actually split into two highly divergent lineages. And those are very largely geographically constrained. All of the ones of higher elevation in mountainous areas in northern Mexico and parts of West Texas and New Mexico were one distinct evolutionary lineage, and all of the ones in central Texas, South Texas, parts of Oklahoma, and the panhandle of Texas, those were a second evolutionary lineage. And so this is really interesting because it actually makes sense with life history. Life history is actually the best predictor here of what's going on. If you look at these areas, the ones in the southwestern clade there, those occur in places where the rains hit very hard. The monsoon rains hit in July and August, which is when most adults are getting ready to pupate and emerge and do their adult activity period. And so that's when most of the rain is happening. These are desert species that can only live in areas when it's recently rained, so they can be brought out by those monsoon rains. Now, if you go to the lineage that's found in the more central Texas area, we see there's actually very little rain in July and August, and the strong rains come in September and October. And so the biggest difference is life history. When they emerge from their pupal chambers is the best predictor of relatedness in this particular case. There was a strong divergence between all of the ones, the colorful forms, which we'll call the C. ledipennus clade, and the other ones, which we'll call C. pollichula. And so there was a major difference in life history. Now, when we used 34,000 SNPs to create a nuclear phylogeny for this group, we found, again, this was a really interesting thing. All of the ones in central Texas and Oklahoma, Cicindela pollichula, were in fact, not only were they different from the other subspecies, they weren't even closely related. They're actually in a completely different part of the tree. And so this similarity of life history and morphology, the differences in life history and the similarity in morphology were actually very interesting here. And we look at the other lineage of all the other colorful subspecies, and they formed their own clade, which is, again, very divergent from the ones in central Texas. Now, one thing that you can do to try to understand better the relationships amongst individuals and populations is to do a structure analysis. So this is based on Pritchard et al. 2000. It's a really excellent way to get a good handle on population structure. Basically, it's a Bayesian clustering algorithm that looks at individuals. Each one of these lines is an individual beetle, and it tries to assign them into populations based on patterns of gene flow, minimizing deviations from linkage disequilibrium and Hardy-Weinberg disequilibrium. And so it's trying to put them into these different clusters. And when we do this delta K method, which actually allows us to figure out what is the ideal number of clusters statistically supported, we find that the nominal species, the ones that were named, are all really well supported, except, again, we find that all the ones living east of that certain line that are found in central Texas and Oklahoma were very distinct from the ones that were found in northern Mexico through parts of New Mexico and Arizona. So that was a really interesting difference that we found there. There is one other thing to be noted here. Two different subspecies of the species Rufiventris were found to share some gene flow, which is not surprising because they're not distinct species. So this actually validated their existence as subspecies. Now, when we go to our mitochondrial tree, it's actually incredibly different. The pattern here is nothing like we saw in the nuclear tree. There's some broad similarities, as in the pollichula clade comes out as largely monophyletic, but there's some weirdness. There's some other things that are inserted in here, things that phenotypically look like another species altogether, but they have a pollichula mitochondria, which is really weird. Why is that happening? We see Rufiventris looks normal, Ledipennus clade looks normal, and then down here, everything gets extremely weird. What is going on in the Cicindela schaupii and its close relative lineage here? There's just incredible mixing. And so despite the fact that these are all easily identified using the nuclear genome, They're not coming out together in the mitochondrial genome. So, why do some individuals appear to have the wrong mitochondrial DNA? This is an interesting question, right? And so I'm going to kind of walk through what's going on here. It's basically a pattern of hybridization and what we call introgression. So when you find habitats where two species co-occur because their microhabitats overlap, you find places like this where there's limestone outcroppings, which is what Polychula loves, and then there's hard-packed limestone sand, which is what Shalpai loves. And the two of them will be found right around each other. And if you're out there in the field, you'll see both species commonly occurring together. Maybe 1 out of 1,000 of them will look intermediate. You can find hybrids. They're extremely rare. They're good at not mating with the wrong species, but once in a while, they make a mistake. And so if we take this and we say we have a Polychula male and a Shalpai female, you can find hybrids. And hybrids will have exactly intermediate characteristics. They have the texture of a Polychula. They have the lack of sutural punctures of a Polychula. They have the hair on the pronotum of a Shalpai, et cetera. And so we found these, and again, I think this is a really interesting example of how introgression works. So let's just walk through this here. So let's take our Polychula male and our Shalpai female, and we have our nuclear genome. Your mitochondrial DNA you only get from your mother. And so when you have mating going on, you're going to get hybrids will have exactly 50% of the genome of both Shalpai and Polychula, but they will have 100% the mitochondrial DNA of the Shalpai. Now, when you backcross this, Polychula is a more common species, so it's very likely this hybrid will backcross with Polychula. You're going to get 75% Polychula, 25% Shalpai, but yet still you have the Shalpai mitochondria that cannot be changed. And so you see where this is going. After just 3 generations of backcrossing into the Polychula gene pool, the progeny will be 94% Polychula. You won't be able to tell that it ever had any Shalpai DNA in it, very likely. But the mitochondrial DNA will remain Shalpai. Now, this is only after 3 generations. After 100 or 1,000 generations, hybridization could have easily happened hundreds of years ago, thousands of years ago. This would be something that you would have a basically 100% Polychula with a 100% Shalpai mitochondria. So that's why we get that weirdness. It also means barcoding doesn't work here. We clearly cannot do that. Because tiger beetles are conservationally important, many of them are rare, many of them have been proposed as species for listing under the Endangered Species Act, and some of them are listed. It's been suggested, well, we should go into areas and we should barcode them, we should barcode the ground, we should do environmental DNA, and we'll know where the rare species are there. It won't work. It simply won't work because of what I just showed you. And so going back to this, to kind of wrap this up here, we have, all this phenotypic variation. And what we learned is this one here in the upper left is the only one that's different, and it's completely different. It's a cryptic species because of its life cycle. It occurs only in the fall when the rains happen. Everybody else occurs in the summer when the rains happen. So life history predicted things well. Phenotype did not. Mitochondria did not. And so trying to come up with objective and repeatable ways of describing, circumscribing species is something that we'd like to be able to do. And in a recent paper that I published in Zoological Journal of the Linnaean Society, we suggest that trying to come up with a congruent analysis of 3 or more data sets out of 5 would be strong justification for calling something its own species. And so these are all relatively independent types of data. And we could look at, for example, a mitochondrial DNA genealogy. Sometimes that gets us to at least species group. It may not be perfect, but it's a quick first pass to see where your major lineages are. We could do a multi-locus population level tree, which is going to get us a whole genome scattershot of what's going on, a population structure analysis. And then we should also include morphology, because sometimes morphology can be informative as long as we're not using really poor characters like color and the white marking patterns, which we've now identified to be very plastic. But using morphological synapomorphies and morphometrics, let's look at the proportions of body structures and how they line up and how that maybe they're statistically different. And it isn't obvious by just looking at the beetles. And then lastly, ecological divergence, looking at differences in life history, looking at differences in habitat preference. And so, for example, in this group here, which is probably the most challenging group to work on, this is the genus Dromaeosaurus. There were 3 species identified within the genus Dromaeosaurus in the past, all of which were extremely similar. And there were only believed to be different because of geography. And so the question was, are these things even different? Are there ways to tell them apart? What's the genetics say? Well, when we did our morphometrics, we found that even though it's hard to tell what these different species are, you can actually look at ratios of the different body structures and come up with ratios that could be used in a morphometric analysis into a principal component analysis. And so what we found was things that were physically not easy to tell apart, we couldn't tell apart when we did morphometrics because they were statistically distinguishable. And so taking all this together, the three species that were nominally three species in the past, we actually found to be 8 species. So we more than doubled the diversity within this group by using this method of 5 different semi-independent data sets and looking for divergences between those. And so we found all kinds of interesting stuff here. And what we found was occasional hybridization in 2 of the species, otherwise largely distinct. And all the species could be identified as distinct based on ecology. Morphology generally distinguished them, but at least 2 of the species were not possible to be identified using morphology, but they were for 4 out of 5 other data sets. And so this is, again, a great way to be able to say, well, we're not going to split and we're not going to lump. We're going to come up with something that hopefully will get us an objective and repeatable way of identifying lineages. Now, in the process of doing this research, actually, we found some really interesting stuff that we weren't looking for. And this is the beauty of science, right? Is very often you find things that you were not seeking, but those are sometimes the most interesting things. And so when we were doing this research, regularly we would find ant heads attached to the antennae, the mandibles, the maxillary palps, different parts of the insects. And those ant heads were identifiable by an excellent ant expert at Arizona State University, and she was able to tell us what all our different ants were. And each one is a record of a natural history encounter between a tiger beetle and an ant. And why are there ant heads attached to them is because ants are vicious, noxious organisms that don't get eaten easily. When a tiger beetle runs up and tries to eat one, very often in their death throes, they will clamp their mandibles onto part of the tiger beetle. The body will get eaten, and it has a permanent earring now that's stuck to it. And now we can see what that species was. And what we learned was that actually quite a few of the ants we were finding were Solenopsis invicta, which is the red imported fire ant. This is a $6 billion a year pest in Texas alone. This is a horrible invasive species, and they're eating them. And so far, no native insects were known to frequently feed on red imported fire ants. So this is an incredible find. This is a several billion-dollar problem. And so what I wanted to do was determine whether or not tiger beetles had eaten red imported fire ants by looking at their gut contents. Now, you can't look for hard parts. You can't look for head capsules because tiger beetles are fluid feeders. They're actually just like spiders. They smash the thing up and they suck the juices out. They don't actually eat all the hard parts. And so I would have to look for stretches of DNA that had unique stretches only found in Solenopsis invicta, like we see here. So here's just a bunch of different ants that occur in this part of Texas. And I was looking to design primers that would be species specific, that would match only Solenopsis invicta and would not amplify in any other ants that they might eat. And we were able to do this. We were able to screen for specificity and make sure it didn't amplify in other species. And there were, in fact, primers that worked. And we found that in some populations, up to 70% of the beetles tested had recently eaten a red imported fire ant. So this could have some applications where we could be doing captive rearing and augmenting natural populations with additional tiger beetles. So I think this is a really interesting thing that we learned. Okay, so lastly, I'd like to talk to you a little bit about rare species in conservation. So tiger beetles are a type of insect, of course, and there's about 1 million species of insects that have been described, and well less than 1% of those species are actually considered to be critically endangered or threatened. And this has very little to do with how many insects are actually endangered and more to do with the fact that we don't like insects and we don't tend to study ones that aren't pests. And so we really have just scratched the surface of insect conservation. It's a very challenging group to work in. If you work in bird conservation, 25% of birds are either conservationally important or soon to be. Mammals, same thing. Amphibians, it's like 35% of amphibians are considered to be endangered or threatened. So insects, it's not indicative of what's really going on. But tiger beetles are actually a great group to study conservation because they're one of the pretty insects, and people like pretty insects. We don't tend to conserve anything that isn't pretty. We can argue how wrong that is, but that's what it is. And so things that get conserved are things like the Carner blue butterfly, Heinz emerald dragonfly. There was actually just the other day a hearing on whether to list the monarch butterfly for Endangered Species Act listing, and that did not go through. And then we have an endangered tiger beetle here, Cicindela oloni, that only lives in a single remnant prairie in Santa Cruz County, California, and nowhere else in the world. It has an address. It's smaller than Rowan's entire campus. So this is a really rare species. Now,
Dr.
one of the
Duran,
reasons—
I just wanted to interrupt for a second to let you know that you have about 7 minutes left for your research portion.
Awesome, good timing. We'll be right on time. So this here is one of the reasons why a lot of tiger beetle habitat exists, is because of fire, right? Fire opens up habitat, creates early successional habitats, and fire suppression has been a huge problem for conservation of not only tiger beetles, but many rare flowers, many other insects, some birds. And so what we see is we're losing a lot of our tiger beetles because of fire suppression. And that surprises a lot of people, but it's not surprising to the conservation community. One of our ones that's particularly adapted to fire-prone areas is the ghost tiger beetle that I showed you before, Elipsoptera lepida. This occurs in South Jersey, and I just published a paper on this, where we're looking at their historical population presence, where they are now, their genetic connectivity, and what can be done about their habitat loss. And so to give you an idea why these things are so particularly sensitive to losses in habitat, they have what's called a shifting mosaic metapopulation structure, and that's how they live. And that basically, I'll describe it as this. All of these little tiny areas of open sand that get formed because of fire burning through an area, or wind, or some other process that opens up a sand dune. When you have that, those habitats tend to be pretty small. They may be big enough to support a population of these breeding, but they're not giant. And so those would only be able to work. They were connected to other nearby patches where they can move back and forth. And if one of them winks out, they can be recolonized. And so they're much more stable as a metapopulation than any little tiny subpopulation. So what happens is, if you put a housing development and an agricultural field and a strip mall, and you take away some of that habitat, you've now created isolated subpopulations that actually have very little ability to reconnect and are much more likely to go extinct due to stochastic processes. And so you really, really want to see connectivity. This is important. Now, we went and we did a survey of all the historic populations that were known in South Jersey. Some were on the coast and some were inland. And we found that these were the only populations left out of about 90 historical populations. There was only about a dozen left. And some of those represent metapopulations that have some genetic structure and some flow between adjacent populations. So this one up here, for example, this is the large reservoir, and these are little satellites to that large set of populations. And we wanted to know what was going on. Where was the best habitat? Where were the places where we had potential for future conservation? And what we learned was the best habitat was man-made. So this is really interesting because of the way that they're doing sea beach monitoring and management in Wildwood Crest, New Jersey. They actually created the perfect habitat for Elipsoptera lepida, because they had these drift fences that were perfectly spaced just apart to create this excellent dune habitat. And they kept areas around it very open, like as if a recent fire had come through there. And we were seeing larger numbers in these man-made habitats than any natural habitat. So it was great because we were actually able to say, maybe we can do more of this in the future to increase the numbers of this rare species. So with that, I'm going to acknowledge some people for their help here. I don't have time to go through all this, but a lot of specimens for analyses were lent to me, especially by a few people at the Smithsonian. Floyd Shockley, Terry Irwin, and Sharon Micheli. Terry Irwin unfortunately passed away last year. Very sad. Many people helped me with field work all throughout all different parts of this project. I'd like to thank some of my co-authors on the Genes paper and the Linnaean Society paper, especially David Herman, who did a lot of the field work and a lot of the lot of discussions. And a lot of photos were taken in this presentation. Some of the most beautiful photos were taken by Jason Sepka and Ted McRae. So with that, I'm just going to talk to you briefly about how I got here, because a lot of times family and friends say, "Why do you do this? Like, what made you decide to study insects and specifically evolutionary relationships amongst insects? That seems really crazy." And it started because I grew up in the Atlantic County coastal plain, um, where there was this one little vernal pond near my house, and it had all these crazy insects in it. I remember being a little kid and just going up to the water and seeing these things swimming around and hunting each other and just being blown away by this whole little universe that was happening in this pond. And I think a big part of what really, really motivated me was right after I got super into it and started to learn what all the different kinds of insects were, it got turned into a housing development. And that really, really hit me hard because again, why do I do what I do? Part of it is conservation, right? I'd like to save these lineages of life that are here and have been here for millions of years. I'd like to see them continue to exist. And this really impacted me greatly when I was a kid. I grew up in the edge of the Pine Barrens, so I got to find all kinds of cool things in the woods. And I went to Stockton University, where I, had a great campus with all this awesome natural forested area. And I decided to study beetles because I just thought insects were cool. And I did a senior project where I looked at all the tiger beetles in 3 different habitats using pitfall trapping methods. And I was finding this one species that was supposed to be incredibly rare, and I got hundreds of them in my traps. And I talked to one of the world's experts on tiger beetles, and I'm 20 years old, so I'm nervous to talk to this guy. And he said, "What are you finding in your traps? Your advisor told me you were finding something cool." And I said, "Yeah, I'm finding Aptrodella unipunctata." And he said, "Yeah, I've only ever seen a few of them. What have you seen?" I said, "About 200." And he said, "I don't think there are that many in all the world's museums." And it blew my mind because I'm like, I'm 20 years old. I just found something the world's expert never found on tiger beetles. That's what he studies for a living. And it just inspired me to say, well, I guess I could find other things. I mean, who knows what else I'm going to find if I get into this. And so, and he encouraged me. He was great. Howard Boyd. I'm actually trying to name a new species after him. So he died a few years ago, but he came to one of my professional talks. I was very happy about it. And, but it was really inspiring. And then, I decided to go to University of Missouri for my master's in entomology because I knew at this point I was going to be an entomologist. And I worked on a completely different group of organisms. I worked on Western corn rootworm because I figured I need to get a job. So I might not get a job working on tiger beetles, but I got to have a backup in agriculture, something that I could do, if taxonomy doesn't work out. And so that was my plan. But I never gave up tiger beetles, and the whole time I wanted to work on them. And I was contacted by a pretty famous scientist at the Natural History Museum in London, Alfred Vogler, and he wrote me an email and he said, "I hear you're going to be the next big thing in tiger beetles." I have no idea who said this to him. I have absolutely no idea who said this to him. And he said, "How'd you like to come and work with me at the Natural History Museum?" And I'm 22 years old with no attachments and absolutely no reason to say no. And so I moved to London. It was super scary. But one of the most important things about that is, you know, I decided to take a risk, and move to another part of the planet. And, I know a lot of people, I talk to a lot of undergrads and they're like, "Oh, I don't know if I want to go to a school that's more than 3 hours away," or, you know what? Awesome things happen sometimes when you take a risk. And so I'm so glad I went to the Natural History Museum and met Alfred, and that's where I learned all my genetic techniques, which I knew nothing about before I went there. So that was a great experience. Um, I worked at the Smithsonian Tropical Research Institute because I came up with a new way to extract DNA non-destructively, and they wanted to hire me to do some work, and it was great. And I got tired of being called doctor when I wasn't doctor. So I kept traveling around doing all these fun things, pre-doctoral fellowship, but then I decided I would go back and get my PhD at Vanderbilt, and I did, and that was great. And since then, I was hired at Drexel University, where I worked as a teaching professor for a while. And now I'm at my favorite institution so far, which is Rowan. It's great. I'm tenure track professor here now and getting to do really cool stuff and kind of living the dream. So that's it.
That's wonderful. Thank you so much for sharing both your research and your career path. We're going to spend a little time now asking some questions that people have been posting. So let me just remind everyone that if you have any questions, please post them to Dr. Claude Krummenacher, and he's helping me organize them, and I'll be asking those questions now. So you feel free to ask questions about the research, and also feel free to ask questions about his career path. And we'll be asking both of those kinds of things. So let me have a timer. Okay, here we go. So there's lots of questions. I don't think we'll get to all of them, but let's just start off here. So lots of different kinds of questions. So given that color patterns can be so phenotypically plastic during development, could they be an indicator of health? Do we know whether females use these traits in sexual selection?
I'll answer the first part first. There's good indication that females can't see most of these colors. So that's the short answer to that part. Is there's a whole lot of stuff that goes into that, but recent studies have said they can't distinguish. And in fact, even shape is a poor indicator. Apparently, there's a lock and key mechanism on the mandibles that is believed to be a big part of mating with the right species and being able to tell potentially how healthy the male is how well he can hold on. And holding on is based on this sort of lock and key. These mandible teeth fit into grooves on the female's neck, and if he can hold on long enough, he gets to mate with her. And if he can't, it's over. She tries to kick him off the whole time, and if she can't kick him off, it works. So, but as far as what was the first part of that question?
The second part was, well, it was since the color patterns are phenotypically plastic, can they be indicators of health?
They're indicators of age in many cases. They actually change color as they continue to dig down into the ground and come back up. They abrade the microsculpturing of the surface, and it actually makes the colors more dull and often makes them more violet shifted as well. So you can often tell when you get to a population, you'll find ones and you could say that's an old individual. It's kind of neat.
Wow. You talked a lot about the phenotypic plasticity and also about the hybridization. It sounds like that might have there been a lot of misidentifications because of those kinds of things through the years?
Yeah, I mean, there are tons of misidentifications. One of the actually most interesting things that happened to me when I went to the Natural History Museum in London is I knew the morphology of tiger beetles really well. I had been studying it for a few years, and I got there and Alfred was like, well, you're an expert on tiger beetles, so you'll appreciate our collection. And I'm looking at his collection and the number of mistakes that were this is the rock. This is the taxonomic rock, the Natural History Museum of London. There were so many mistakes. And I was very sheepish at first. I said to him, I said, Alfred, a lot of this is wrong. And he's like, really? I said, yeah, a lot of these things are misidentified. And he said, take the next 6 months and fix everything in the collection. Just go through and just fix everything. And it was a major undertaking, but yeah, mistakes are very common because a lot of species overlap in color and pattern. They're usually different, but you have to look at different characters if you really want to be able to identify them. There's characters that work better than color and pattern, and everybody wants to use color and pattern because it's easier,
Yeah. So let me just switch over to a career question for a second. So how old were you when you were working in London?
22 years old.
22 years old.
No, sorry, 24 years old.
And there you are, pretty young still, correcting like the world's rock, as you said. How did you feel like being that person who was knowledgeable enough to fix what the experts had done?
It was intimidating and awesome. Like I was really, I kept thinking like, really, am I the one who's going to do this? Like this has been, this collection has been here for hundreds of years, and who knows how long some of these mistakes have been, embedded into these drawers. And, but I don't know. I loved it. It was awesome. I absolutely adored my time at the Natural History Museum. It was fantastic.
That's neat. So let me just play off that for a second. There are so many species of insects and groups of insects in the world. I feel like, students at Rowan or other places, if they wanted to specialize in something and become the world's specialist, there are a lot of different areas they could go into. What do you think about that?
Oh yeah. I mean, so there's almost 200 families of beetles, right? And there's about 400,000 species of beetles. So there's more beetles than anything else. And I mean, probably 50 of those beetle families, if you were motivated, you could just be the world's expert in that family. You know, like it's just, if you really decided like, I'm going to do this group here. I was actually talked out of being a tiger beetle expert because when I briefly for about a year and a half gave up tiger beetles and said, when I was at the Smithsonian Tropical Research Institute, I worked on a project on leaf beetles, and I was told there are thousands and thousands of people that study tiger beetles, and there's books written about them, and there's all these. Why are you're not going to be the world's expert? Like, do something else. Be more pragmatic. And I couldn't give them up. And the whole time it was like talking about your ex. The whole time I'm working on leaf beetles, I kept saying to my boss, I'm like, oh, it's really cool because tiger beetles do something really similar. And he's like, all right, just go work on them, man. You got to do that. It's crazy that you're working on anything else. And I mean, and you could argue at this point that I'm the world's expert in tiger beetles. So I don't know. But you know, I didn't give up on it. I just didn't give up.
That's great. That's great. Thank you for sharing that personal note. So another question was, you mentioned that life history differs between beetles, and you mentioned measuring ecological divergence for circumscribing species. Can you say a bit more about the life history traits in this group? So how long is the lifespan? How many times do they mate? Do they migrate? If they do migrate, is it different between males and females, and how far? Stuff about like who their predators are and kind of competition.
Yeah, no, that's a great question. Some basic things are, when are the adults active, right? So that's a really important thing. They all have a period of a few weeks when they're most active. And that could be in the spring, that could be in the midsummer, that could be in the fall, that could be after rains, that could be in the dead of winter around permanent water. If you're in a subtropical area, it's really going to diverge a lot in different groups. And then a lot of it has to do with the larvae. We don't talk about the larvae that much, but the larvae have complicated life cycles where sometimes they actually overwinter for 2 years to get enough food, and then other ones go through in 1 year. You know, there's all kinds of behavioral differences about whether when they fly, do they do zigzags or do they fly in straight lines? Like, there's all these different things about their natural history that you know, you could look at. So lots of stuff about them, and more all the time, because very little has been done on tiger beetle mating. Like, it's weird how little we know about it. We know a few things, but relatively little.
You mentioned that some species, like the larval stage, will be 1 year and some will be 2 years. Do we have any that are like fairly wide distribution north to south, who, like, some living in the south might go through 2 cycles during a year, or just a difference within species?
No, absolutely, 100%. The big difference comes down to obligate diapause versus facultative diapause. Do they have to overwinter as a larva, or do they have the ability to push through if it's warm enough? Some of the species, it doesn't matter what it is, they have a spring fall life cycle, and they estivate during the summer. They just hang out in the summer underground, and it doesn't matter what latitude you're at, they do that. So, and other ones, it's like you said, if you go to like Alaska, we have species that make it all the way up to the Arctic Circle, and they take 4 years to finish their life cycle in Alaska, whereas they might take 1 or 2 years in California. So
That's neat. Thank you. So a lot of people were interested in those hybrids, and they were asking questions like, are the hybrids better or worse at survival? Or do we even know that yet?
we don't know. Almost the hybrids are seen so rarely that we know almost nothing about them other than their genetics, because after they're dead, you can still get DNA out of them. But nobody's ever done. I mean, I've seen in my entire 20 years of working on tiger beetles, since I was an undergrad, I've seen 4 hybrids all over the world. 4 times, 4 instances ever. Most people have never seen one. So they show up in collections every now and then. And then I have a friend who found like a giant hybrid swarm in this one place where like hundreds of hybrids were occurring. But I mean, that's once in a lifetime stuff. You just don't see them very much. So
Wow. Neat. Thank you. Does the time in which the beetles are hatching affect their phenotypic variation?
yeah, it probably does, because Timing is going to relate to weather, and if it's basically if it's more humidity and it's cooler, you're blue shifted or
Mhm.
violet shifted. If it's
Mm.
drier and hotter, you end up red shifted.
Right.
So yeah.
Wow. That's so neat. Thank you. Um, so what are you expecting the effects of climate change might be on the life history or other distribution of our tiger beetles?
Well, that's a great question. And actually, Dr. Andrew Garner and I are going to be working on a study together looking at whether or not species ranges have changed between 1900 and 1970 and 2000 to 2022 to see whether there's been any kind of shift in their ranges, like a southern retraction and a northern expansion, and specifically like what kind of temperature changes were enough to cause those.
Mm,
So we're going to look at a big data set to do this for all 230 different taxa in North America. So
neat. Thank you. Wow, that sounds like a really large project.
yeah, it's part of a grant that we're submitting in the next couple of weeks. So
So will you need some Rowan students to help you out with that?
actually, I'll need 3 Rowan students for sure. Yeah.
Excellent. Um, so some questions varying around some different ideas of are people, are there any researchers who are actually raising these in the lab? And if you could do that, would it be possible, like if for the rare species that are endangered, would it be possible to raise them in the lab and then release them out into the field?
Yes, and very yes. Um, one of my closest colleagues, who's a co-author on 2 of the studies that I showed you, Roger Gwiazdowski, he's at University of Massachusetts, and he has been rearing tiger beetles in the lab for 10 years, or actually it's probably closer to 15 years now. And he is actually doing it for the federal government. They're taking rare species that are nearly extinct and they're captive rearing large, like thousands of them, and
Wow.
releasing them into reclaimed habitat that was previously destroyed and has now been reclaimed,
Wow.
with mixed success. Sometimes it works really well, and sometimes for reasons that no one can seem to figure out, they just disappear. They don't take. We don't know why.
Hmm.
But that's something that we're looking into.
I know that with some vertebrates, there are sometimes behavioral changes when we've reared them and then released them compared to their wild counterparts. Is there any, do we know enough about their behavior to know if there would be a difference between the wild ones and the reared ones?
No, that's a great question. And because they're scared of that being a factor, they release them as third instar larvae so that when they're out, they'll put them back into the ground and let them come out as adults on their own, just in case releasing
Wow,
adults doesn't work the same way. So yeah, it's a great question.
that's neat. Thank you. What would you say are the biggest threats to the tiger beetle populations today? Um, and what can we do about that?
I mean, habitat loss is number one for sure. I would say significantly greater than climate change. Not that climate change isn't an issue, but I mean it just there's a tiger beetle in Miami, the Miami tiger beetle, well named, that only occurs in an area of a few acres left in the whole world. It's right near the
Um,
Miami Zoo, and they wanted to build a Walmart on top of that area, and just trying to stop those things. I mean, it
mhm.
was emergency listed. I was part of the federal panel that determined that that was should be listed as an endangered species, and but it's just tough. It's just housing developments are killers.
Mm, mhm. Are there ways to perhaps reconnect isolated populations? If there's any work toward that or progress toward that?
Um, I mean, I hope. um, it's hard, because corridors are hard for a lot of wildlife. Sometimes they work for certain groups, other groups they don't. Tiger beetles are very vagile, meaning they disperse really well. So if you have habitat that's reasonably close to another patch, they usually do find it.
Mm,
They can fly. An adult beetle can fly at least 30 to 40 miles in one season. So we know they can do it. Actually, we know that there's a species that's been found 300 miles offshore on oil rigs at lights. They're attracted to light, and they were found 300. It's a species that's found sometimes on the wrong continent. This is an incredible dispersing species. Think of it as a bird, because it's just they show up in the middle. They're a coastal species, and they've shown up in Kansas before.
Wow.
They just disperse. Yeah.
Wow, that's fascinating. I'm going to switch over to some career questions because we have a number of those being posted. And some kind of big picture questions here. How would you describe to lay people how your research is useful to the world?
Well,
non-scientists.
Sure, absolutely. We have lots and lots of data that says that biodiversity is keeping us alive. There's a thing called ecosystem services, which says the more species, and we know that the more species that are present in an area, the better the ecosystem services are, meaning you get clean water, you get clean air, you have crops grow better. All kinds of things happen when you have more biodiversity present in an area. And this is not contentious anymore. So, I think biodiversity is important. I think species have a reason to exist and have their own right to exist. But even from a selfish point of view, who knows? Again, look at the tiger beetles that I say. They may eat fire ants, and they do eat fire ants, and so that might be something that we could do. So I think that biodiversity has a lot of value.
Yeah. Thank you. Yeah, absolutely. A couple of questions about your London experience. How did that shape your research and your field studies? And if you hadn't really had this opportunity to take that risk to go someplace else, how do you think your career path would be different today?
Oh, that's a good. Well, to answer the first part. It's where I was 24 years old, and I was trained in classic ecology and entomology. I was not a molecular systematist at all. And so it's where I learned to stop being afraid of genetics. I'm like 24 and I'm like, get over it. And and then I really learned a lot of those techniques from Alfred, and many more on my own later. But I think it was a really good segue to that part of my career. And then what would I have done if I hadn't? I have no idea, but I'm sure it would be very different than what I'm doing now. It's very hard to say what I would be doing, but there's a good chance I would be in agriculture. There's a really good chance I would be working in agriculture, which, has its own reasons for being, but just, I don't think that would have been what I wanted to do the most.
Although I did see on one of your, I think it was LinkedIn, a mention of looking into effects of industrial hemp.
Yeah, that's a new project that I'm actually interested in as well, which is very, very early on. So we're just getting started with that.
Is that more of a this would be habitat loss, or is that more of a here's a new species being planted and what are the effects on tiger beetles?
So that would be more of a, are there ways to grow hemp that maximize biodiversity, and so that increase pollinator numbers rather than decrease them, and things like that. So basically restorative type of agriculture.
Awesome, thank you. Excuse me. If students are interested in studying abroad or doing research abroad, what advice would you give them for both how to go about getting that, and then while they're there, maybe overcoming some of the fears and the homesickness and things like that?
It's look into everything, I think really a big part is just look into, when I was applying to PhD programs, when I had my master's, I had all this experience in London, I had all this experience in the Smithsonian in Panama, I just went far and wide and said, where am I going to do the next step? And, Vanderbilt seemed like a great fit. I found a, professor who really wanted to work with me. If I'd only gone and applied to a couple of schools, maybe I would have gotten into them. But, I just think it was really good to kind of make sure I had checked all the options out there. And, getting over the challenges of something that's new and scary, just I think you have to think about what a cool opportunity is and keep that in your head the whole time, because I had some really bad experiences in London too. I almost left. I really did. I mean, I was living in a very, very, very rundown area. I had bed bugs in my apartment. I was mugged twice. Um, I mean, it was a rough, rough, rough place that I lived in, and because I could afford it, and that's all I could afford at the time. But everything got better, and I just, I kept saying to myself, if I do this, this could be like the whole rest of my career. I just have to stick it out. And I was actually able to do that in 24. I really was. I was able to like try to step back and see the bigger picture. So
Yeah, it's important. Thank you. Good perspective. So a couple of questions about what part of your career were you at when you discovered the 11 new species of tiger beetles, and how did that feel? And why were you able to do that when it seems like other people weren't?
So awesome question. Okay, let me think about this. All right. So the first one that I discovered was in Panama. That was in 2005, but I didn't name it until 2013.
Hmm.
So now I generally do things in a much quicker turnaround time. But I was still in my 20s, and there was other priorities, and I found it. I had become such an expert on Panamanian tiger beetles that I knew when I saw it right away that this was unnamed, because I knew what everything else was, and this wasn't one of them. So, um, you know, but I use a combination of field work, like going to remote places. That's what I did. I went to a remote mountain range in Panama that nobody goes to at a time of year that nobody goes, looking for a group of beetles that nobody was looking for, and so I found something new. But a lot of it comes down to doing genetics for me now. Like I'll go and I'll say, all right, this is a big group and there's some weirdness going on. Some of these things seem pretty different. They have different life histories in different places. I'll bet they're not the same species. And then you do the big genetic study and you find, oh, wow, every time that they have this life history trait, they're different. So, there's different ways to discover things. Sometimes I discover them in museums. At least 2 new species I've discovered in the last year. There was one in a museum and it said indeterminate. It was just unclear what it was. It's all it said on the label. And I looked at it and I said, oh, wow, I've got to start doing some measurements on this thing. This thing is really, really different. So, you find them in the field, you find them in museums, and you find them with genetics. You find them different ways. But almost all of them were described between 2013 and a month ago. So, yeah, recently.
Wow, wow, that's neat. Thank you. One last question, when we have a little bit of time left. Would you talk about the influence of the varying aspects of what's been more valuable, maybe? Your career, your education, or your experience? How have those influenced?
Wow. That's a good question. I really have to think about that. Clearly, it's a strong combination of both. I can't figure out which one is going to win out here. It's sort of like you need to be good and you need to be lucky and you need a lot of things to go right. And I think experience has been hugely important for me. But I couldn't have done this without getting a PhD from Vanderbilt. I mean, I wouldn't have gotten a job at Drexel as a professor if I didn't get a PhD. So education is critical. I mean, if you want to do research like I do, you're probably not going to be able to do it with a master's. You're probably going to have to go on for a PhD. And I like all the institutions I went to. I thought about them very hard before I made those decisions. So it's hard to say, but I mean, obviously, experience is hugely important as well.
Yeah, yeah. All right, thank you so much. I wasn't able to ask all the different questions that students were asking, but I appreciate all the time that you took to respond to these. So I'd like to thank everybody for attending. Dr. Rand, if you could stick around for a minute at the end, but thank you everyone for being here and for asking so many good questions and participating. And just a reminder that we do have a number of seminars coming up and we invite you back again. Thank you very much. Goodbye, everyone.